Hello, folks. This is Steve AB4L. I was going to mow, but it started to rain. The weeds will have to wait until at least tomorrow, right? Now, nothing in this episode is a promotion, nor is it a recommendation. Just going to let my mind wander. Let's see what we find out under the covers. See, that's the point of letting your brainstorming and letting your mind wander, is there's the hype on top, the headline, the clickbait, and then there's the real story. Facebook has 3 billion users. Many of them are old. Well, I'm not one of them. Nintendo, I don't play games. Apple, I don't have anything Apple. I eat apples for my health. Gene editing makes bacteria-killing viruses even more deadly. That almost sounds sinister. So this is an abstract from Ars Technica, and it's by one of the regular highfalutin abstractors. You know, Slashdot is news for nerds, but there's a couple of different guys that pull stuff in from all over, and they have what appears to be a privileged situation where I don't think the ones they submit get voted on for mainstreaming. That's a guess. This guy's one of them, because I've seen his name posted by like 1 million times. And there's a lot of other ones you only see him once or twice. So an anonymous reader shares a post. So somebody posted this, but didn't want to use their name. Or they're registered as anonymous, and this guy picked it out and promoted it. There we go. So you just got to let your mind wander and brainstorm a little bit. Broad spectrum antibiotics are akin to nuclear bombs. Obliterating every prokaryote they meet. Now, if somebody else wrote this, then they do have a science background, biochemistry, microbial genetics, animal genetics. In other words, you wouldn't write it that way. And I wouldn't write it with that tone. They're effective at limiting pathogens, sure, but they're not so great for maintaining a healthy microbiome. What's my microbiome? Intestinal flora. Actually, at my last appointment with my GP, I actually used the phrase biome, and she said, "You mean flora, don't you?" Don't switch me to these highfalutin new phrases. Ideally, we need precision antimicrobials. Microbials are bacteria, fungi. Do we want to hold yeast as separate from fungi? Spirochetes, are they separate from bacteria? They can target only harmful bacteria while ignoring the other species we need in our bodies, leaving them to thrive. Now, I've seen in the past year a couple of references to some medical studies where there are people who simply have 100% diarrhea 1,000% of the time. There's something happening in their whole alimentary tract that they're unable to actually, when you poop, those are bacteria. but they serve a very important process in the digestion and of food and modification of certain things, getting it ready so that it can be absorbed into your bloodstream and your body to use it. Your body does things too. So what they've done with certain really cases, I think people who are on the verge of death, they've taken the shit from healthy people and put it in there. And in some cases, that's what was needed to get them back to normal defecation, growth of the biome, microbiome, flora in your gut, and save their lives. And you might go, ugh. And no, they didn't sterilize it first. The whole point was to put it in live the way it came out of someone else, because that was the magic bullet, not drugs. All right. See, my mind wanders. So, enter Sniper, SNIPREBIOM, a Danish company founded to do just that. Its first drug, SNIPROO1, is currently in clinical trials. The drug is designed for people with cancers involving blood cells. The chemotherapies those patients need can cause immunosuppression, along with increased intestinal permeability, that means injuries, and so the bacteria, which are usually mostly E. coli, seep into your bloodstream and begin to spread. And yes, if they colonize in certain susceptible organs, I can think of someone in my lifetime who died that way. Can cause immunosuppression along with increased intestinal permeability, so they can't fight off any infections they might get from bacteria that escape from their gut, their intestines, into their bloodstream. And then they go on to talk about how this company does things. They were talking about why the antibiotics are not useful. So then it says many of the infections are caused by E. coli, and much of this E. coli is already resistant to the typical drugs. They started by screening 162 phages. You know that word, don't you? To find those that would infect a broad range of E. coli strains taken from people with bloodstream or urinary tract infections. And then they settle on a set of 8 different phages. You know what phages are, right? They engineered those phages to carry genes that encode the CRISPR. Never mind what that is. That's a recent technology that's finding expanded use. You're born with defect in a certain gene and you're homozygous for it and you're going to die. And especially in people who they know are going to die, they are experimentally going in for certain of these defects with the CRISPR system in a baby and modifying certain target cells that multiply and become organs to correct the genetic defect and to make them normal. And these kinds of experiments being done so recently directly on humans is because the cases they're selecting are going to die soon, based on 100 years of medical knowledge and experience. I'm explaining to you why certain things might be done to a baby as an example. If you go, isn't that terrible? They're experimenting. No, that baby's going to die. Not sort of maybe. And you'll find there are certain other very leading edge, cutting edge experiments done with CRISPR and other things on people who are at the edge of death. So what's there to lose? Even they consent. What's there to lose? I'm days from dying. But you know what these phages are, right? Then they talk about how they modified the phages and they're packaging for them together that they've selected. They don't make a drug. The team confirmed that it remains stable for 5 months in storage and does not affect other gut bacteria. So these phages, which is a shortcut word for bateriophage, these phages specifically target E. coli. So if I just go like this, what is the phage specific to E. coli? Protection of bacteriophage-sensitive E. coli, PNAS, Proceedings of the National Academies of Science, National Academy of Sciences, pardon me. Protection of bacteriophage-sensitive E. coli by lysogens. So here's an article where they're trying to preserve the E. coli that are in your gut. But if they get into your bloodstream, they can kill you. And if your immune system can't handle them, all right, I'm reminding you of what are lysogens? Something that will break up something else. What's a lysogen? Dictionary, a lysogenic bacterium or bacterial strain. A lysogen or lysogenic bacterium is a bacterial cell which can produce and transfer the ability to produce a phage. See, they're using the word phage again. A prophage is either integrated into the host bacteria's chromosome or more rarely exists as a stable plasmid. What's a plasmid? It's a circular DNA, not a part of the chromophore. What's the chromophore? Chromophore is the chromosome of a bacterium. It's a different word. It has a different structure than the ones in, well, people like us or trees or mice. But in the abstract level, it performs the same function. So what's a plasmid? It's a circular DNA that gets replicated right along. Oh, wait a minute. Oh, that's where the foreign genes land in the plasmids that know how to kill antibiotics and make that bacterium resistant to drugs. Oh, that fits right in. This is the first time I'm going through this, folks. Here's a nice image. You see this guy right here? Let's see if they have the picture here. Yeah, let's see if we can get it big. See this guy here? That's a bacteriophage. And what's this here? Bacterial host in lawn. They're growing bacteria there, and they have sprayed or plated bacteriophages in there, and then they get this clear spot, and that means that there was a successful infection. And they go through and explain what all of this is about and how the bacterium then produces more bacteriophage to go and sit on and then inject DNA into the next bacterium. Those are called bacteriophage. Well, oh, is this something new in nature? When were bacteriophage discovered. And did it get a Nobel Prize? National Library of Medicine, The Strange History of Phage Therapy. Since the Enlightenment, I'm just letting my mind wander, folks. Half of this is new to me. Since the Enlightenment, scientists have enjoyed a self-image as rational actors. That's got a tone to it just like the one that wrote the thing in /dot, but a different kind of mindset. Guided only by reason, evidence, and logic. When the Royal Society of London was founded in 1660, it chose a motto, nullius in verba, that's Latin, on the word of no one. Now, what does that mean? I say it's that way, so you have to believe it. That's what they're against. Show me the evidence that I can reproduce. You may be the big commie boss or the president of the United States, and I'm nobody, but just because you say it doesn't mean it's true, even though you're trying to make me swallow it. I'm explaining what they were up against in 1660. If you were to take a look at what the situation was in Britain at that time, you'd probably get a sense of why they were revolting publicly, politely, in a way where they wouldn't get guillotined or hung. Soon after Felix Darel discovered bacteriophages in association with diarrheal illnesses. Isn't that what we were talking about before? People who have diarrheal illnesses. He speculated that phages were responsible for unusual recovery from such diseases through their antibacterial action in vivo. Oh, you mean they were like an antibiotic? The people who had this bacterial diarrhea and that if you fed them something with a bacteriophage that could attack them, that you could cure the person? What year was this? I've never seen the name before. Let's find out who this person was. I'm just letting my mind wander. He looks French. He looks like 1890s. It's a guess. At least there was photography. Born in 1873 in Montreal. Oh, he's a Quebecois. Born in Montreal in 1873, died at age 75 in 1949 in Paris, France. So I was right about the French part, too. He was a French microbiologist. He must have immigrated. He was the co-discoverer of bacteriophages. He also was an applied microbiologist. That's what that lab is about. All right? With his money almost gone in his second daughter one, he took a contract with the government of Guatemala as a bacteriologist in General Hospital in Guatemala City. So he moved around a bit. And then in 1907, he took an offer from the Mexican government to study fermentation. He moved to Yucatan, Mexico. And then he went back to France, and then Argentina, and then back to France. Never read this article before. That's a bacteriophage. Where's the genome? Where's the DNA? Can you take a guess? In the head. What does this do? These pincers are the thing that are site-specific receptors that can allow it to grab onto a recognition site on target bacteria. And what happens down through this once this penetrates the outer skin, it's not the right word, of the bacterium. It injects what's in the head through that into the inside of the bacterium, and it replicates itself in there, and then finally it destroys it, and not one but multiple then go free to go and find some more to do it to. In a certain way, what recent pandemic was handled by a mechanism similar? See, there's only a few common themes in biology. I'm just letting my mind wander, folks. Who won Nobel Prize? I don't know if I spelled it correctly. I did. For phage mediated transformation. Now these are all they showed me the bateriophage thing again. Phage it's interesting to look at. It's interesting to look at the way the search engine can't see past 6 months ago. Who discovered bacterial transformation? And I'm going to go DNA. Question: who discovered the phenomenon of transformation in bacteria and in which year? Oh, wait a minute. Let's use oh. Wait a minute. Griffith's experiment, reported in 1928, was the first experiment suggesting that bacteria were capable of transferring genetic information through a process known as transformation. These findings were further research in the 1930s and early 1940s that isolated DNA as the material that communicates this genetic information. Then they talk about Spanish flu. And I'm still looking for the person. The exact nature of the transforming principle of DNA was verified in experiments done by, that's 1944 and 1952. Do you see the bacteriophages? And they call them bacteriophages. Now, why'd I go off on this? They're using something that in certain scientific terms is ancient. Who's they? Who's they? Clinicaltrials.gov, a study investigating the safety, recovery, and pharmacodynamics of multiple oral administrations of Sniper 001 in healthy subjects. Why would they do an experiment like that? To prove that it's innocuous to healthy people. Don't do it on sick people. Give it to somebody who has no problems. And if they don't get sick, if giving it to someone who does have a problem that it's targeted for and they still die, it wasn't because of the medication per se, due to the toxicity of the medication per se. Did you follow that? Would you think to do that experiment? The safety and scientific validity of this study is the responsibility of the study sponsor investigators. A listing of the study does not mean it has been evaluated by the U.S. federal government. Read our disclaimer for details, blah, blah, blah. It says it's active but not recruiting. First posted March 14th, 2022. So it's been under, I'm hitting the wrong button. Come on. Last updated December 2nd, 2022. Sponsor Sniper Biome APS. Collaborators Wellcome Trust, that's often involved in health research. What does it say here? This is phase 1, randomized, double-blind, placebo-controlled, multiple-dose escalation study in healthy participants investigating the safety, tolerability, recovery, and whatever PD is, pharmaceutical dosage, of multiple oral administrations of this agent. Condition or disease: E. coli infections, bloodstream infection. What two kinds of things are they going to administer to people? The drug, Sniper 01, and a placebo. What's a placebo? There's nothing there. It's inert. Why would they do that? To filter out people who have psychological reactions with physical manifestations. Phase 1 suggests that there'll be other phases, but this is the initial one. Let's look at what the trial is about. Interventional clinical trial. Actual enrollment, 36 people. This is last year. Allocation randomized. See, back in the late 1980s or early 1990s, statisticians started to go through the medical literature, pardon me, of Europe and North America. And with the kind of computers that you had, they began reanalyzing published, accepted, peer-reviewed medical trial literature, often taught as examples in medical school education curricula. And what the statisticians who were not physicians and had nothing at stake discovered and proved. Remember what the Royal Society motto was? Never mind, you're saying, here's the proof and you can reproduce it. Half of the published medical literature was false in its conclusions. What are the results and what's the discussion about why we did this, what we observed, what we got as numbers, what's our conclusion, what should you do with this? half of it was wrong. It should have been the opposite statistical conclusion. We say this drug cures this type of cancer. The analysis of the medical literature of the Western world said half of what's been published that had supposedly a statistical analysis, the analysis was wrong. And it should, in half of the cases, flipping a coin, folks, it literally turned out to be, in about half the cases out of thousands, it should have been the opposite conclusion. And they teach it in medical school. They were teaching it in medical school. What's the point of clinicaltrials.gov? You can't do anything today without having submitted a complete, thorough experimental design that follows recognized statistical procedures and can be tested and fall back in case a certain amount of your patients fall out of this study. At what point will your study no longer be valid? Don't bother continuing if you lost a certain number of participants. And it all depends on what the design is. And the design is randomized, sequential assignment. Would you like to sign up? The first one gets assigned over here. The second one gets assigned over, but there's certain categories. So sequential but randomized. They didn't just say, oh, we've got 36 and we'll take the ones on the left side of the room. They didn't do it that way. Dose escalation, masking. Oh, this is so that nobody can react or say or do anything because they think they're getting the real thing or not getting the real thing. Quadruple. The participant doesn't, the patient doesn't know what they're receiving, the drug or the placebo. They don't know, and no one's going to even hint at it. The care provider, that's the nurse, the person at home, the person in, they don't know either. The investigator, who's the investigator? That company. How it gets assigned and to whom? Yeah, we've got all these vials, half of them are placebo, half of them are the drug, and we hand them to someone else who takes care of deciding who gets which. And I don't tell you, the drug company, what I did. I keep it a secret. I lock it up. And what's the fourth one? The outcomes assessor. Remember what I said about the statistical analysis going back over the literature? Why would the person who's the outcomes assessor, they have to take all of their data and send it to somebody that they don't know who it is. They're reputable, they're professional, they do it. You don't know their names, you can't influence them. I watched a video yesterday. It was a rabbi that works in the United States, and he's a kashrut rabbi. He goes and he inspects places of business that produce food in one form or another in order for it to be certified as good kosher, good for, you can eat with a kosher meal. Who do you think he's been the rabbi doing that for, the kashrut, for something like 30 years? Coca-Cola. And what the humorous title of the video was that the actual formula of what goes into Coca-Cola has always been a secret. Supposedly it's locked away in a safe, and there's only like 2 people in the world in Coca-Cola that know what the actual ingredients are. And the rabbi even said the way they actually do the manufacturing steps, no one gets to see the whole picture, but he does. Because in order to certify it as kashrut, he has to be able to know every single ingredient, step, handling, place, go and see it, everything. But he's sworn to secrecy. Only he and Hashem know what he knows. That's what it means by outcome assessor. He's like the kashrut rabbi. Masking description double blind. It's more than double blind. Phase 1 randomized double blind, first in human, dose escalation study. They want to start with a small amount. And if there's no fallouts from sickness or anything like that, in any of the people, then they like double it and then double it, whatever. They want to see how much a new human can stand, and these are people who are healthy. Yes, you have to do this with drugs. Dose escalation investigating the safety, recovery, and pharmacodynamics of multiple oral in healthy subjects. They expect to complete the study on April 30th, which is over now, 2023. So I was wondering, who are the people in charge of it? They go into more. It says Sniper 1 is a live biotherapeutic product consisting of genetically modified bacteriophages. They don't say phages. Bacteriophages, specifically targeting Escherichia coli. And I'm trying to find informed consent, male or female, healthy subject, no known clinical relevant organ abnormality, disease, blah, 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 no clinically significant abnormalities between 18 and 65, E. coli present in the feces sample. Yeah, they actually check your poop before they start and during the trial. Normal defecation pattern at least once daily. It's right there. Willing to participate in the study and provide fecal samples. You shit every day and you send it to them. Or they pick it up. And then there's other things like you're not HIV positive, you're not pregnant, and all that kind of stuff. And it's being done in Ohio. But the company's in Denmark. Right? So if we just do Sniper Biome, a Danish company, whoop. There's Bloomberg. I'm not promoting anything. It's news. We are pioneering CRISPR-based, that's gene editing, microbial gene therapy, nonprofit patent licensing platform to enable the field of CRISPR editing in prokaryotes. News and press. Nature Biochemistry, Biotechnology, Science Report, DK, Biocapital, Lee. They have more than 40 scientists, but I'm looking for a particular one. I'm trying to find the names. Give me a name. Who's the scientist? Who are the actual faces? Who are the actual faces? What did I Oh, look at that. This is where they got the article from. There's the bacteriophage. The researchers showed that Sniper 1 was well tolerated in Göttingen mini pigs. After oral administration, the pigs did not exhibit any clinical. Oh, so they experimented on pigs first? I'm not finding the name I found before. I'm looking for the scientists because, just like when I do the hacker stuff, I like to know who the scientists are. Are we going to have any names? Copenhagen. They say who the CEO is. Oh, there it is. Article citation, Nature. uBlock has prevented me, I say proceed. Nature Biotechnology. Nature magazine, British publication from, Royal Society, something like that. So I was looking at this. Engineered phage with antibacterial CRISPR-Cas selectively reduce E. coli burden in mice. So this was a research paper. This would be proceeding human trials proving that they can do it in mice. They can do it in pigs. We're ready to try it in humans. And I'm looking at the names of the scientists. Now remember, I'm looking at the names. Now this is Denmark. Wait a minute, I can download the PDF. Let's see if I can download the PDF. No, I don't want to do it that way. Hang on. What are they doing to me? I'm looking for, I can kill this. That helps. Everybody wants to push. Get in your face. Microsoft, Mike, you should read the stuff about Windows 11 where Microsoft is jamming the ads in now. That was one of the top things on Slashdot today. They own the world. So I was interested in who the hackers are. Just like I do with the... And I looked... See, I want to make that a little bit bigger. I'm looking at the first name. Now it says that the authors mentioned here, all the way up to Anders Ostergaard Peterson, all contributed equally. Usually when you see an article, the first scientist's name is called the corresponding author. And usually if it's multiple, that's the person who, it's either it was the only one that was willing to take the time to write it up because it was a disaster, or this is the person who organized it and everybody basically submitted the results to that coordinator, who may also have been a principal investigator also. But I'm looking at the names and it says the whole first bunch of them. Now, like that last one, Anders Ostergaard Peterson, I can see that as Danish. This name right here, I can see that as Danish. Virginia Martinez, that doesn't look like that's Danish per se. And this one here, Szabolc, is that Danish? Camille Robert, that sounds like that guy's probably French. So this may have been an international cooperating study. Or maybe they all work in Denmark. Maybe people flocked to Denmark, which had a very good COVID period because of the way they handled things, which was very different than what was done in Great Britain and the United States. So I was interested in this one. Look at that name. Isn't that beautiful? And I'm asking myself, where is that person from? And with an A on the end, I assume it's a female, but I could be wrong. Senior scientist, PhD, ResearchGate. Hang on. See, I'm just tracking down the hackers now. And she works for that company, PhD in Health and Medical Sciences. She's got 15 publications. Google runs something called ResearchGate, which attempts to abstract and put in one place all of the scientific research papers to let you know where they are so you can jump to find them instead of going to the library. I don't know. Are they going to show me the LinkedIn? Let me try an incognito page. Now, the first thing I'm noticing, it's not in English, but I can see the word profile. And DZIU must be her family name. That's a guess, but maybe it's her first name. I can't tell. I can't tell. Senior scientist at Sniper Biome, a CRISPR company. Now, I like this. Do you see the way they spell Copenhagen in Danish? Can you see it right there? Copenhagen in Havenstad, Denmark. She's got 431, around 430 followers. This guy, oh, there's the other guy over there, the one I was wondering about that name. We'll check on him. And there's that guy too. So they all work at that company. She says I'm a highly motivated, this is just the same thing I do with the software hacking. I'm a highly motivated microbiologist, we want to advertise ourselves, with an interest in different fields of research, including molecular diagnostics, food microbiology, infectious diseases, and antimicrobial resistance. I am a team player and have experience in multidisciplinary collaboration. Well, you often have to be multidisciplinary if it's a medical study. So the question would be, where'd she go to school? Because that'll give us a hint about her country and her name. And what languages does she speak? That's what I do with the hackers, isn't it? So now it's in English here, Copenhagen, capital region, Denmark. And then it switches to be Danish. You see, the dates are in Danish, but they didn't use Danish here, but they used Danish up there. Did you see it? And she's talking about 3 years and 8 months. Senior research in the Copenhagen area, postdoc at the University of Copenhagen. Okay, that's how she got into that company. That's how she found it. That's how they know about it. She did her doctorate in Copenhagen. And you meet a lot of people that way while you're doing your doctorate because you give presentations and you also often have to have cooperation from other people in other labs and sometimes outside cooperators. Research assistant, National Veterinary Institute, Technical University of Denmark. So that goes along with the experiments on the pigs and the mice. Research assistant, National Food Institute, Technical. Okay, so she, before she did this stuff, got her PhD, she actually worked, so she went back to school. Or she was supporting herself for some reason before going back to school or at the same time. PhD student. Okay, so she worked as a research assistant, not a postdoc. PhD student from 2009 to 2015 in University of Copenhagen. And then she was a technician research assistant in Vilnius University. And that explains the name. She's from Lithuania. She's Lithuanian. She got out of Lithuania and went to Denmark. All she had to do was swim across, right? I'm not making a joke. You know what I'm talking about, don't you? Let's get a map. So here's Denmark, right? And there's Lithuania. You see it right there? Whoop. Can I make, oh, let me scroll it up a little differently. There we go. So my head's not in the way. There's Lithuania. This is part of Russia. There's Poland. And there's Denmark up here on the peninsula. So she could have taken the train, but she could just as well have swam it. You think I'm kidding. When the Germans, the Nazis, formulated the plan to deport all Jews in Denmark because they had invaded Denmark and proclaimed it a protectorate, as an example of what being under the Reich's care would be like. A German businessman at the embassy heard talk in the embassy about rounding up the Jews, and he went to an office in downtown Denmark, to the resistance office. Yeah, the Danish underground resistance had an open door policy in downtown Copenhagen. Yeah, the Nazis knew where they were, but they didn't mess with them, because periodically the Danes would do something like at 1 p.m. on a Monday, everybody suddenly comes to a standstill. All the traffic stops, nobody moves. Even when the Germans put guns to their head, nobody moved for, I don't know, a minute, 2 minutes, just to show that 100% of the population was against them, against the Nazis. And then they would go back to normal activity after the 1 minute. All, everywhere in Denmark at the same moment. Which I suspect is one of the reasons why on Heroes and Martyrs Remembrance Day in Israel, all traffic, everything, everybody on the sidewalks, cars, it doesn't matter. For 3 minutes, 2 minutes, 3 minutes, we all stood in silence as the sirens blared. Same thing. So anyway, the point about the Danes was they decided to round up their Jews and take them by fishing boat to Sweden and dump them there, so the Nazis couldn't get them. Anyway, so I'm saying maybe she swam from Lithuania to Denmark. Oh, we haven't finished. Which one is hers? Wait a minute, I lost track. That's ResearchGate. Is she over here? Which one is hers? Did I lose track of it? Is it in the other one? Oh, there it is. I found it. I'm getting old. PhD, molecular bacteriology and infection. Vilnius. What are her languages? Let's see if we got it. Let's see if she tells us. SPOG, her languages: English, professional, practical, Danish. I'm assuming it, let's see if that means native. I understand when it says professional, it means she can read the scientific literature and she can communicate in a note that way. But that's not the same as conversational. So if we copy this and we open a tab and we go to translation and we do that, limited practical skill. So her Danish is limited practical skill. Well, but let's get but her English is professional practical skill. How could she have limited practical skill and get a PhD in Denmark? How could her Danish be limited practical skill, but she got a PhD in Denmark? Did that occur to you? No, she doesn't mention Lithuania. She's from Lithuania, but she doesn't mention Lithuania. What does that mean? It means I'm never going back. I don't know her, but I've known other people. That means she's never going back. But how could she get a degree, a PhD, without being really fluent in Danish? Did that pass your mind? Now this is ResearchGate. Postdoc in University of Copenhagen, research assistant, show all. University Milan, researcher, Vilnius University technician, the Italian thing for, just half a year. These publications are in English. Publications in English. They don't translate it for you. If this was in Danish, it would be shown here in Danish. These are the abstracts. So they were published in English language, scientific English language journals. So we're going back in time. That's 2013. Qualification of salmonella in pasteurized egg products by some form of PCR. Conference paper. She's been a steady researcher, 2008. There's her network. Doesn't mention her PhD. Self-sufficient cytochrome P450 monooxygenases from Bacillus thuringiensis. Amplification and comparative sequence analysis, 2008. Could you have said all of that? So she's been in the field for a while. Now, I'm curious about something. I'm curious about this guy here. He's the lead right there. I'm curious about that. All right, it's a guy. It says DK, Google DK. Senior scientist, verified email at bacteriophages, phage-derived proteins. This is Google Scholar. It's got a different layout, but it's .dk, not .com. Can you guess at what his name is from? I'm guessing it's not from that first name. I'm guessing that it's not Danish. How to pronounce National Institutes of Health, Turkish. Italian. But these are papers published in other. LinkedIn. Turkish Coast Guard. Somebody with a very similar name right here is Turkish Coast Guard. So indirectly, we figured out where he's from. So we got a Lithuanian that swam across and a Turkish. Well, he could still swim there, across the Mediterranean, through the Straits of Gibraltar, up through the English Channel. Are they going to show me anything? They don't want to show me anything. Is it slow? I killed hers. It's not going to show me anything? Doesn't want to show me anything. It's in Turkish. So do you think that's a good guess? Do you think that's a good guess? I think, yeah, there's another one here. It's another name that's very similar. It's also published on a Turkish government site. Look, this is called international teaming. What you do is you get the best minds you can. You don't ask, how did you get here? I'm interested in this guy too. Let's see where this name is from. This is just like me figuring out who the hackers are. Sometimes they're all from Israel, and sometimes they're from like half a dozen different countries. Google Scholar, ResearchGate, University of Copenhagen, LinkedIn, Team Lead. Let's see if they'll give me an incognito page. See, the problem is, now let me show you what it is. What do you want to bet I can get it? We're going to copy the link address, and I'm going to open a different browser that already blocks them knowing who I am. Brave browser. You see what happened? Did you see what happened? See, I'm a hacker. There he is. That's his picture. We saw it before. Let's see if we can find... Oh, I'm afraid we can't see much about him. He doesn't want to talk. Oh, wait a minute. No, I'm wrong. Denmark, Budapest, Hungary. Quality assessor, contract, National Institute of Pharmacy and Nutrition, Hungary. Assistant professor at some university I can't pronounce, Budapest, Hungary. Visiting fellow, Bethesda, Maryland, National Cancer Institute. Staff scientist, Hungary. What are his languages? These are his degrees. Yeah, he got his master's degrees in Hungary. He got his degrees in Hungary. I once had a major professor who went across the bridge during the Soviet invasion of Hungary to get out of the country. Andow, is that right? Bridget Andow? Did I get that right? Boy, that was a long time ago. Is that how you spell it? No, that's not how you spell it, but that's close enough. The Bridget Andow is a 1957 nonfiction book by the American author Michener, chronicling the Hungarian Revolution of 1956. I said I once had a major professor who crossed the bridge at Andow to get out, when he and his wife to get out. Let's see what his languages are. English, Hungarian, and Danish. You notice that he mentions the Hungarian and she doesn't mention the Lithuanian? I'm not getting down on her. He's going back someday. I don't think she is. I think she's shooting for somewhere else. Bigger than Denmark. After she makes her reputation. And a project like this one that we've been talking about, a project like this, if it succeeds, definitely. This could be, if their strategy is good, and if there's no subsequent biome adjustment, this would be terrific. And it would be a project that you'd be happy to say you were a part of. Steve A. for you, just letting his mind wander and doing a little bit of brainstorming too. Saying see you in 73.